And now today we are here at 2025 with lots of building, but still we are falling short of space because the way we are expanding. And what are the activity we are doing in this small space is that ILS, whose mandate is for the betterment of human health and welfare, with the objective that it's promoting the basic and applied research in frontiers of life sciences. Broadly, the science that we do over here with about 26 faculties and technical staffs and other administrative staffs and about 150 plus students, PhD students and project assistants also. We work in the area of cancer biology, interdisciplinary biology, infectious diseases, and plant and microbial biotechnology. Of course, like a sinusoidal wave, these fields, the number of people working in this area go up and down depending on new recruits and scientists retiring. However, these are the four major focus area in which the scientists and the groups can be grouped into. We have a good amount of facility and very cutting edge facility even in this small space. As you can see, most of these facilities are available through a platform called Sahaj platform, where people, where faculties and researchers from the city and from other places.
Can you please unmute the mic? We are not able to hear.
Really?
It's heard here. I think you have some audio issue. Please try.
Oh,
no, we are not able to hear.
So there must be a local issue, sir, at your end.
Even from ICGB.
We can hear in ICGB. No problem.
Yeah.
So ICGB, Trivandrum, all people confirming that they can hear. So I'm sure anywhere there is an audio issue, please look at your local audio issues.
Yes, we are able to hear. From NIBMG, we can also hear.
Thank you. Appreciate it. So coming back, so in terms of facility wise, it is quite cutting edge, and not only the ILS researchers, but people from outside. Can you please mute somebody in the online platform? If you are not asking a question, please mute your mic. I'm sorry. Can the administrator also take care of this? Okay, so moving on, this is about the facility. Now let us talk something about the technologies that are developed here from the research that has carried out over here. So, fulvin as an adjunct drug for the treatment of cerebral and severe malaria. So it is a FDA approved drug. I'm really sorry. Administrator.
NCCS can mute please. NCCS.
HDMI be unplugged.
Can you please?
NCCS
yes,
can
we
you
can
please
hear
unmute?
Yes. Since I am seeing the full screen over here, I'm also not the administrator. Okay. Thank you. So this is a very interesting story coming out of ILS. Dr. Arun Nagaraj and team has worked on a FDA approved drug, which is actually approved for antifungal drug. But now that we are seeing, it is working wonder for cerebral and severe malaria, and we are quite hopeful that this drug also solve quite a bit of malaria issue, which is endemic in Odisha's tribal regions as well as Africa, and many other countries are facing this problem. The second story, which is about multi-drug resistance, a formulation which can inhibit the growth of multi-drug resistance. Dr. Tushar Beoria and team has worked on it. They have utilized FDA-approved drug, which has the efflux pump inhibitor to provide resistance against multi-drug resistance against staph aureus, which is an incredible story. So these are few technology I just named. I'm not going to take more time on this. There are few startup companies that have started at ILS. There are many, but the three that I mentioned over here are actually from the faculties of ILS. So Polyorbit Private Limited by Dr. Mamoni Das, RNA Biotech Private Limited by Dr. Amaresh Panda, and last but not the least, Dr. B. Ravindran, who retired from ILS as a director of the institute. He also started a company from the science that he has worked done at ILS. So it is about nitric oxide producing agent, NOCIP antagonist. So these are all individually interesting stories. I hope some other time we'll get to discuss about details on each of these scientific things. There are a few products also that have been developed at ILS. Magnetic nanoparticle based cell separation kit may sound that it is trivial, but no, this there are requirement by the faculty, by the researchers, and this was seen by one of our startup companies, and they said, okay, why not we create such simple magnetic nanoparticle based cell separation kit. And then we also have antibodies for chikungunya virus. Dr. Soma herself has spearheaded this work. And RNA biotech, the one I mentioned just now by Dr. Amaresh Panda, also looked at the day-to-day need of the research laboratory and then tried to prepare kits, which is working really wonders, and it can challenge many established companies because the prices can be lower and the efficacy is better. And there are many companies, as you can see the logos on the bottom right, which are not started by ILS faculties, but then people over here, they took advantage of the ILS Bioincubation Center. So in the last 5 years, the bioincubator has supported about 44 startups. 51 IPs and trademarks have been filed. Nearly 10 crores plus fund has been raised, as well as many products, 69 products commercialized, 250 people have been employed, 147 programs going on, so on and so forth. So, in fact, the BioE3 policy has made our life easier because employment, environment, and all these things, economy, we are already working on it. So this is going now to take a good pace over the years. Here is a glimpse of few products that have been developed at the bioincubator, and they are in the market. The wellness fragrances, neuro cosmetics, so on, and this is my favorite one, which gives a lot of happiness by drinking these beverages, instant tea, so on and so forth. Many things are there. I'm going to end my this talk with a initiative that has been taken here at ILS. We have a natural strength or advantage of having a large coastal line, more than 500 kilometers, largest brackish water lagoon, rich marine bioresources, about more than 31% of Odisha has a forest cover. 1/3 of the state is in the hilly region, and several tribes are there. So given all this biodiversity and all, we thought of entering into a new regime, new program, which is called Bioprospecting Resources from Odisha's deep ocean. So where we are trying to get into not only Odisha, also southern part of India, the peninsular region and the Bay of Bengal, we are collecting samples from at various depths of the sea. But we have confined ourselves to the river estuaries where fresh water comes and meets with the ocean. And we are collecting samples from 0 meter, 10 meter, 20, 50, 100, 200, and from the Bentham. What we are trying to get, look at the various microbial species that we can get. We're trying to isolate compounds, a lot of interesting molecules that we have got there, and which we are trying to look at the applications there. So in a nutshell, if I can give a glimpse of what all things we are planning to do, we are looking at the biodiversity of marine microbes, multiomics of the marine microbes, bioprospecting those for human health and industrial application, developing an AI/ML pipeline for the data analysis, Odisha has recently started a program called BRIC. This is not the BRIC that DBT has started, but this BRIC Odisha Marine Biotechnology Research Innovation Corridor, where the institutes located in Odisha, either Berhampur, Fakirmon University, Berhampur University, NIT Rourkela, all these people now engaging into a top-down research where the ILS is going to play a crucial role of having the biorepository and providing genomic solution. We are planning to start an enzyme production program and a biomanufacturing hub at ILS with these focuses. I don't know how much time I have taken. Maybe I have overshot a little bit. These are some of the ILS outreach programs that we have conducted in last 2 years or so. So not only the research that I spoke just now, which is deep research, we also try to educate, excite the young masses from schools, colleges. Our faculties go to remote places where people cannot come here due to logistics issues. Then the faculties go there and then excite them about prospects of biology, biotechnology, and similar area. So this has been showcased on various occasions in the Indian International Festival, Science Festival, and National Science Day, building entrepreneurial ecosystems. So that's all from my side. I think we will now go to the next piece of program. Can you? From DBT, is it okay that we move on, or if there are any queries or any questions that anybody want to ask, do ask through the chat boxes. That way it can be interactive and we'll try to address. But meanwhile, I think we will move to the next program, which is the panel discussion. Spot on time. So Dr. Soma, you are going to moderate, or you are going to moderate? Subho. Yeah, go ahead.
Good morning, everyone. It's my pleasure to welcome you all to this very special panel discussion as a part of World Bioproduct Day 2025, organized by DBT and hosted here at BRIC-Institute of Life Sciences, Bhubaneswar. We are honored to be joined by three distinguished scientists of ILS. First, I am going to Dr. Sanjeev Sahu. Sir, for your, I want to ask you that what is your opinion on the potential of Odisha's coastal region to contribute to India's bioeconomic growth in the near future?
Yeah. So as you know, strategically, Odisha's location next to the Bay of Bengal, it gives a pleasure, So that, and also we have a 480 kilometers of, coastal belt with a diverse ecosystem with rich biodiversity. From Gopalpur to Diga, we have the Chilika Lagoon, and also we have a lot of mangrove forest, near to Tarkanika. So it gives a very high value added phytochemicals for manufacturing, for pharmaceuticals, cosmetics, and also the presence of this seaweeds and seagrasses. Also, it gives a additional value in the case of pharmaceutical industry.
Thanks, sir. It's very encouraging to hear that the local marine ecosystem can feed to the national level bioeconomic strategy. My next question to you, based on building on that what are your views on the use of marine resources in pharmaceutical research, particularly in the context of cancer biology?
Yes, as you know, chemotherapy basically used in case of for the cancer patients. But due to the high toxicity, the alternative, they are looking for some phytochemicals which has a non-toxic nature. And I am very sure, lots of work is going on. Since the phytochemicals constituents from these seaweeds, seagrasses, and also the mangrove leaves. Due to that non-toxic nature, maybe it will be used as a supplement or value added to the existing chemotherapy for better treatment for cancer patients.
That's truly inspiring that the idea might someday lead to the next generation cancer therapeutic. Now I'm going to the Dr. Shoma Chattopadhyay. Dr. Chattopadhyay, what are your views on the use of marine resources in human health to research, particularly in the development of antiviral and antibacterial agents?
Yeah, so regarding this antiviral research, till now there are not many compounds in the market, but there are a lot of publications where macromolecules or like microorganisms of marine origin has been shown to be a potential molecule, like from red algae or cyanobacteria from sponges and many other like plant also, even seaweeds. It has been shown that for HIV, for SARS, even dengue, different arboviruses, as well as for herpes viruses, it has been shown that they are showing antiviral property. But this has not come as a product till now, but it needs a lot of other effort to bring it to the market. But yes, there are potential.
Thank you, ma'am. The emergence of antimicrobial resistance certainly highlights the need for new naturally derived compounds, and marine microbes could be the gold mine for that. My next, just shifting the gear slightly, that what is your opinion on the role of marine agriculture and marine derived foods in adding the nutritional needs of growing global population?
Yeah, I mean, marine ecosystem is relatively, less explored, and my biodiversity anyway is a big potential for our country. With this long coastline that we are discussing since morning. So whatever we have discovered till now, we have many things to be explored, and definitely that will give a huge source of food, not only for human being but for the feed for the animals as well as for aquaculture, and definitely that will boost the economy of the cultivars in and around the coastal area.
Thank you, ma'am. And you already introduced the marine algae like seaweeds, and also next I'm going for this similar type. Another question, which is not the application now, is little bit of basic biology and systematics of understanding our marine ecology. So now I'm going for Seema Pradhan. Dr. Pradhan, is there a role for basic biology, particularly genomics, in advancing our understanding of marine algae?
Yes, I believe there is a very big role for genomics to play. As Dr. Soma very rightly pointed out, there is a huge biodiversity which has not yet been explored. And I believe that the first step to exploring this biodiversity should be a good foundation of genomics. That will provide us the data about what are the biomolecular markers which can be used for identification, diversity assessment. And also, since the concept of junk DNA has become almost obsolete now, almost all part of a genome, even the non-coding part, has a critical role to play. And in case of seaweeds especially, there is very little genomic data available till now, which is a huge potential for exploration. So there is novelty in the research which attracts many people. And also there is a huge scope for application. So yes, I think genomics would be the way to go for a basic biology approach.
Okay, absolutely. Understanding the genomic architecture of marine organism is a key to unlock the full potential. And I'm glad you emphasized that. So could you elaborate on how genomics can be applied to enhance seaweed cultivation or improve the production of bioactive compounds?
So we can go by two parts of this question. So first was your question on the application in seaweed cultivation. So in this context, since we know very little about the life cycle of seaweeds, what they need to grow, there is very little input into the seaweeds. They grow very fast. So we can use that area of exploration. We can explore what makes them grow so fast and apply it for seaweed cultivation, improvement of the cultivars. Then the second question was about improvement of the bioactive compounds. So I'll give an example here. The main product of seaweeds is carrageenan. It's a very huge potential of carrageenan in a market because it's a gelling agent. Usually it is used as a substitute for gelatin. It's a plant-based substitute for gelatin. So it has huge applications in food, pharmaceuticals, cosmetics. But the demand of carrageenan is so high, yet we have to import all those biomaterials from countries like Indonesia, China, and Thailand. India already has a huge potential for seaweed cultivation, as already pointed out by both of our senior scientists. So it would be a mistake to not tap into that potential and go for a multi-omics approach rather than just the genome. Genome can form the foundation of what we are looking for, and then transcriptomics, metabolomics, proteomics, everything has to go hand in hand to identify the particular genes that need to be manipulated and use it for betterment of the seaweed culture.
And if I may add, I think this panel has rightly ventured into a topic which is currently very hot topic. There is a high-level meeting going on at the secretary level who are thinking as seaweed as a future feedstock. All the attendees of this meeting, particularly, there is a good news. DBT and other ministries are going to come up with a call for proposals where people who are interested in seaweed biology might get a chance to showcase their particular skills in this area. So what you'd have, it would be like a relay race. So for example, how to grow seaweed in huge quantity. So that is a production. The second would be how to identify which seaweed has a more commercial value or more potential for other usages. And then there will be chemical engineers or bioengineers that would be required who can process them chemically or enzymatically. Then they can reduce the oligosaccharides, polysaccharides to oligosaccharides or monosaccharides. And then ultimately, I think the country is looking at it as a top-end game as to how to produce alcohol from this the seaweed or the feedstock or the animal feed or aqua feed. So this is going to be an exciting decade who are getting into seaweed biology. So Seema, well said. I think multi-omics is the way to go forward, not just the genomics, but rather the transcriptomics, the metabolomics, understanding the pathway. Maybe genetic, some editing technology would be required, which can bring it back to things that are already consuming those and not letting bigger or saccharide to form. So there are many exciting questions out there.
Yes. Sir, I'd like to also point out a new area of research here that is into the microbial associations with the seaweeds, because that has been a very unexplored area. The diseases that are affecting the seaweeds, because the production ultimately suffers because of those things. So there is a huge potential for microbial exploration also associated. So I'd like Dr. Shubhobrata to elaborate a little bit about the fungal pathogens and maybe about the endophytes especially.
Okay. So yes, as you said that endophytes is very new in case of seaweed biology, but endophyte are within the seaweeds, and it's a vegetative growth, so it's transferred from seaweeds to seaweeds. So at ILS, we are also looking for the different type of endophytes from different region of the Odisha and others part of the country, and we are excited because lots of endophyte we have already isolated, and we are going for their sequencing and their understanding. And as you know that it's very difficult to spray anything in seaweed because it's in a sea. so it's wipe out. So maybe in the future we are planned or we try to develop some technology based on endophytes which can protect or grow the seaweed and protect the seaweeds in the harsh conditions or different conditions. And that's why we need a knowledge, and we need as much as endophyte we can isolate and collect. So I've also wanted to ask Sahu sir, we have some time. So what do you think that those fishermen, those are depend on the fish, how we can motivate them to cultivate the seaweeds, and others like other state already doing that, but how Odisha can be motivated them? What should we do for this aspect?
Yes, it is a good question, So in Odisha, we have one or two startups like Brahma Kamal and all. They are growing the seaweeds. The main question is, in the rainy season, due to the heavy rain and changing the salinity, they could not do for 3 to 4 months, And once more, when they want to again to grow, they need the, the small part, from outside, which is very costly. In this regards, we have already started at ILS. Can we have a biobank, So maybe we can supply at the same time. At the same time, also, we need a more or less to convince the farmers and give a orientation. So, and at the same time, also we have to prove it is basically economically viable. So I think time is coming. We can do
or
that.
it can be side by side with the fish and seaweed. Both can be cultivated in that way.
Yes.
Okay. Sir, I wanted to ask director sir. So must be there is some policy can be needed, or some specific plan national wise we need to promote the seaweeds. So what is in your mind? How we can start?
Absolutely, I think you are spot on. Policies are needed, policies are there, policies need to be edited. I think this is a topic which requires interministerial discussion. See, when we talk about seaweed, it is just not in Ministry of Agriculture or with Ministry of Forestry, Ministry of maybe Fisheries. So very timely, people have realized. I think there is an interministerial discussion that is going on, that has happened recently, and the key people are talking at the top how to make clearances much easier, so that scientists who are working at the on the seaweed biology, they are not disturbed much. Only the main, I think, requirement would be at the production level, where industries or mechanized industries can easily produce a large quantity of seaweed. But once that is produced, processing them or doing a research, there are no hurdles at this point of time, I believe. But do you all realize that at ILS, not just we are going deep into the ocean, some of our startups also have gone up, taking biology into the space. So,
yes, so our next speaker would be speaker. So as a closing remarks of this discussion session, thanks to all our panelists for such rich discussion. From this discussion, we realize that marine biotechnology is going to be a strong pillar in establishing BioE3 policy of our state and our nation. Thank you, DVT, for this national platform. Now we are going for the next part of our program.
So let's hear some exciting journey.
Yes, I request Dr. Soma Chattopadhyay to for the next session.
So our next talk would be from an innovator. He is talking on pharmaceutical research and how we can do that in space. So Antariksh.
The speaker's name is Antariksh, and he is also working on space biology. So interesting.
Hi everyone. Honored to be part of this discussion. So we are Serendipity Space. I'm the co-founder and CEO. My name is Antariksh, and our mission is to accelerate drug development on Earth by processing pharmaceuticals in space or microgravity. For that, we are building small reusable satellites, which are about the size of a travel suitcase that you usually carry around in your trips, which can go to space, autonomously produce pharmaceuticals and proteins in space and bring them back. And there is a neat story to be narrated here of why do we even need to think about going to space. Yeah. So the current state of all pharmaceutical companies around the world is determined by this staggering statistic, which is that to bring a drug from its birth to its maturity, it takes about 10 to 15 years, costs $2 to $3 billion, and even then the failure rate can be as high as 90%, which means that at any stage the drug development can fail. But when we started with Serendipity, we were wondering what the reason for this statistic is. And turns out that we are not focused on clinical trials per se, but everything that comes before that. And when you think about everything that comes before clinical trials, the first step, which is the discovery of the drug compound, as well as optimizing it and developing it into a formulation, there are a number of scientific and industrial challenges that pharmaceutical companies face. Some of them are you often are not able to crystallize the drugs as well as the proteins, which are often known as undruggable targets. The second is that even if you're able to crystallize the compounds, when you move towards the formulation stage, you often face issues such as you are not able to control the particle sizes of the formulation or the solubility. And all of this actually leads to very low quality formulations, which have to be done with repeated trial and error for years. And this is where, around almost a decade and half the total cost of drug development goes into. And this is where we thought we could have an impact by basically collapsing this time and money that was spent on doing trial and error by doing more focused discovery and development of drugs. And the industry secret has been that over the last 30 years, I mean, it's not very common in Asia or maybe in the East, except Japan. But in the West, companies have often gone to the space environment to bypass some of these problems because zero gravity has no sedimentation, no convection currents, and no buoyancy. So as a result of which net new materials can be produced. And this is not only true for pharmaceuticals, that is also true for metal alloys, semiconductors, and fiber optics as well. So, this has been happening since the last 30 years, and I want to show you some commercial successes that have come out only in the last 2 to 3 years. And you can see that the first set of images over here, which are the blue ones, this is a monoclonal antibody drug made by Bristol Myers Squibb. Now, they were not able to get the drug, like the correct crystals of the drug in the right geometry, in the right size. But once going to space, they actually got much more uniform crystals, larger and structurally better. And the second set of images, which are the black and white ones, is these ones is the most actually the biggest success over the last 2 to 3 years, which is of a drug called Keytruda. Now, Keytruda is the world's highest selling drug. It does about $25 billion annually in sales every year. And, they were trying to make a subcutaneous formulation of this monoclonal antibody drug. However, they, were not able to make it because they had issues with agglomeration as well as viscosity, all of which were solved by going to space. And they brought it back down, and this is astronaut Thomas Pesquet actually mixing Keytruda in 2 bottles in the International Space Station. And what our plan is to build a reusable satellite which is less than 100 kilogram, which can go to space and then manufacture pharmaceuticals over there over a couple of weeks or a month as required, and then re-enter into Earth's atmosphere and then have a soft landing somewhere on the coast or on the land. Now, what is exactly the vision of the company is that we want to start out for the next 5 years to actually produce gram scale crystals of proteins as well as pharmaceuticals to basically, accelerate drug discovery and development. we want to use that revenue to fund the further manufacturing of a larger satellite, which can do kilogram scale manufacturing of APIs such as Keytruda. Now, this unlocks a huge market which was not possible before of pharmaceuticals that actually cannot be made on Earth currently. And we think that our projected impact is going to be something like 20 to 30% cost reduction in overall drug development, as well as a reduction in time to market of a drug by 3 to 4 years. And this is not us saying it. We talk to companies all over the world. We talk to Novartis, Pfizer, Bayer, Roche, and from that we actually, and the companies that have actually done this in the ISS. The reason that they were pursuing this was that they actually expected, and in some cases they actually got drugs which hit the market with much lesser time. And this is our team. We have a brilliant team ahead of us. Jivitesh and Monica, they're handling the technical side of things, and Monica previously worked at NASA as well. She's handling the space side of things, and I'm basically the salesman of the company. We have in our advisors, we have Dr. Subrata Sen, who previously worked at Pfizer India and Syngene as the scientific manager, as well as the director at Aragon Bio, as well as Dr. T.N. Suresh Kumar at ISRO, who has helped us get inroads with ISRO. Now, where we are in terms of our product is we have built a small autonomous pharmaceutical factory, so to say, which can grow crystals of proteins and small molecule compounds, and also do formulation such as crystalline suspensions. And just in the last 6 months, we developed this. We tested it in multiple labs across India, and we successfully produced simple proteins like lysozyme, apoferritin, as well as some small molecule cancer drug compounds as well. And we have been lucky to be a part of BRIC-ILS bioincubator. They have provided us with tremendous amounts of opportunity and visibility, which is why we were able to get some of our customers even in India actually. And so this is our plan. We have already have an agreement with InSpace and ISRO, and hopefully whenever the next launch opportunity comes up, we will be sending our payload to space to autonomously manufacture pharmaceuticals in space as a technical demonstration. That's about it. Thank you so much for listening.
Thank you, Antariksh. It's very exciting research that you are entering into, or you have entered into already. And at the same time, it is very costly also, this kind of research that you are entered into. So are you facing challenges of getting fund or to do this research, and how much fund have you generated so far?
Right. So we have raised our first pre-seed round worth a little more than $500,000,
Wow.
and to basically fund the manufacturing of the core technologies to build out the satellite. And of course, it is going to be a little capital intensive, but I'm pretty sure that the rewards that come out of it will dwarf the investments we raise.
And you told me that you missed the Axiom 4 boss because you started, the research that is going on Axiom 4 have started long back. By
Yes.
the time we were not even.
Yes. So we're less than a year old as a company functioning, and by the time we realized that there was opportunity for Captain Shukla to actually fly to the ISS, the research had basically been decided of what all experiments are going, but otherwise we would have been a part of them for sure.
So, okay, hopefully we see some of your ideas crystallized, and not only proteins also. Thank you very much.
Thank you so much.
ICGB has raised hand. Are you trying to ask a question, ICGB?
We have a question, Abhishesh. I have a question for Antariksh. The question is, the crystals, it's good that they form in space, but when splashdown occurs, won't the crystals disperse? How do you cushion the landing?
Right. So basically, fortunately, there are two things like that have been established over the last 30 years, that the crystals, once they return, they actually retain their form and structure, because the medium in which they return, or like the apparatus inside which they return, it is ensured that it is viscous enough to not crash into the walls. And so the vibrations, the high G effects, everything that is required to be done, the crystals are not damaged. And that's the same logic that we are following as well.
Okay, yeah, thanks. Very nice.
Thanks, Ramesh.
One more question, actually.
Okay.
So there are certain setups where astronauts can train on the Earth, where there is zero gravity and the simulations of atmosphere, which is like in space. So why do we need to send the drug to the space? Instead, can't we work on that in the simulations, because that will be cheaper also?
Right. Yeah, brilliant question. I mean, trust me, if we had the chance to do that, we wouldn't go to space, because going to space is tough. But the reason is that there's a reason it's known as a simulation for astronauts, because the maximum gravity level that you can reduce on Earth by any by any machine like a clinostat or anything is 10 to the power minus 2 Gs. Which is 1/100th of Earth's gravity. But what you actually need for any meaningful advancement in pharmaceuticals is 1/1,000,000th of Earth's gravity, which is microgravity. So 10 to the power minus 6. So, and there is no, the laws of physics don't permit creating a microgravity environment on Earth. So that's the reason that we have to resort to space. Otherwise, I would have personally done it on Earth.
Request everyone to put your phones on silent mode. Request all of you to put your phones on silent.
Yeah. So should we go ahead with the plan, as because one more speaker?
Yeah, you can continue.
Yes. So thank you, Antriksh. If there are more questions, we'll get back to you. But let us go ahead as planned. You are here on this side. Okay. So we have with us. Yeah. So you introduce. So Dr. Vishwaranjan Pradhan.
Yes.
Right. Yeah.
So he is the founder of this organophonics, and many of the students will be really encouraged seeing him, because he is a PhD. He got his PhD from NICER, working with Dr. Polokai, and then he did postdoc at ILS with Dr. Shanti, and now he is having his own startup. Yeah.
Fantastic. PhDs to postdoc to having own startup. It's a very interesting journey. Please share your. You want some slides, huh? Slides. See. You can go ahead while he is projecting his slide.
Hello everyone. So good morning. So so coming to organoponics, so it is actually started in 2021, but the research and the ideas and everything. So it was much before, around 2008. It was been established, because I'm from near Chilka. Okay, so which is the largest saltwater ecosystem here in Odisha, and I have seen that around 9,000 acres around the Chilka is being cultivated with aquaculture, particularly for the shrimp farming, and over 15,000 acres for the fish farming. And so what they do, okay, so coming to the problem. Next. Yeah, just click on here. Yeah. So coming to the problem. So in Odisha, sorry, in India, around 20 lakh acres of area is being used for the aquaculture, which releases around 2,000 billion liters of water, which are actually nutrient rich, to the nearby water bodies. And in Odisha, around 2.5 lakh acres, which releases around 250 billion liters of nutrient rich water to the environment. And coming to the Chilka, so around 264 billion liters from the shrimp farming is being released, and which increases the eutrophication as well as the Phragmites growth in 1987 to 2024. Around 14% increase in the Phragmites in Chilka is because of the nutrition from aquaculture and from the sewage flowing through the rivers from the Bhubaneswar municipality. So we have come with a solution for the both of the things. So the first one is actually which are the microbial compositions. So mostly to recycle the nutrients, okay, which are flowing from aquaculture, from the sewage, as well as from the agricultural fields to the Chilka Lake. So super microbiomics is one such So which is actually a microbial composition mostly for the soluble components, organic components, and STX is for the insoluble ones as well as the regeneration of the soil. And so pro microbiomics, okay, it is particularly for the aquaculture one, okay. So mostly for this continuation of aquaculture and recycling the soluble wastes coming from the metabolism of the shrimp farming as well as coming from the feed and other nutrients given to aquaculture. And for the growth, okay, we are given this organ boost. And particularly the biggest problem is the ammonia nitrate and nitrate, okay, which are generated in aquaculture ponds, okay. So we have a microbial solution called NILSA, which effectively releases, recycles all these things, okay, into the planktonic system, both in the phytoplankton as well as the zooplanktons, and again, incorporate into the food system. So increasing the decreasing the FCR and increasing the productivity. And the major problem in aquaculture around this area, it is an Vibrio. So so we have a microbial composition, okay, which is called Vibdux. So which competitively suppresses the Vibrio growth and suppresses the white gut, white fecal systems as well as the mortality in aquaculture. And to treat white gut, we have another product called Vidux. Okay, so this is again a gut microbial composition. And okay, I have a small video to play. So what impact we have created? So we have enhanced the socio-economic status of over 6,000 farmers in coastal districts of Odisha. Out of this, okay, around 2,000 farmers are from around the Chilka Lake. And we have enhanced the shrimp productivity in Chilka Lake region by 2,500 ton in last 4 years, amounting around 75 crores. And we have a plan for the next 5 years to enhance, particularly from 9,000 acres around this area. So with decreased footprint by around 1,200 crores. And we have treated over 150 billion liters of aquaculture water. So that was directly released to the Chilka Lake, and which has this microbial composition, which can digest the sewage coming from the urban wastes. And around, yeah, so that was the impact created in last 4 years. Thank you very much.
Thank you. Congratulations. From PhD to postdoc to now having your own startup, and you are also working in the area, the theme that BioE3 is biology for economy, environment, and employment. Environment, though I can understand, employment also I can understand. Yeah. I hope very soon also you will have that economy part, which will make you rich and famous. Okay. Great. So I think we should move on with the stop sharing. Okay, with the program. If there are any questions or discussion needed, we can come back after this BioE3 policy, that city place that we are going to take. So what I'm going to do, I'm going to take the place here in the city of Bhubaneswar. And I do not know anybody else who is from Bhubaneswar, Odisha, who have joined through online can take the place along with me and whoever wants to. So you people are ready to repeat after me. It is going to take some time. It's a long place. Let us stand up. Okay. पास पेज नहीं पहुंचेगा तो शेयर इट मेरे बाद रिपीट कर लीजिएगा. Are we good to go? Yes. Ready? Okay. No, he says and then we repeat. Yeah. So the place to advance the BioE3 policy for a sustainable future. Please repeat soon after me. We, the people of Bhubaneswar, in the state of Odisha, wholeheartedly affirm our commitment to the vision of the BioE3 policy of the government. A strategic framework to build a sustainable, inclusive, and innovation-driven bioeconomy grounded in environmental conservation, economic opportunity, and employment generation. Recognizing our state's unique scientific, ecological, and cultural context, and the immense potential of our bioresources, we pledge to implement the BioE3 policy with a dedication and leadership guided by principles and priorities. Promote and actively collaborate on the bioproduct innovation. Unlock local and national potential. Establish and promote biotech innovation hubs that drive a regenerative circular economy. Enable